So I'm presenting this data on behalf of all my colleagues who are part of this pretty large phase 1 trial. This is on bb21217. So it is targeting BCMA, which we're all quite familiar with right now. And what we are going to be presenting at this year's ASH meeting is a total of 72 patients. This was a phase 1 study, so we went through a dose escalation. We started off with 150 million cells and then went to the target dose of 450 million cells. And the difference between the approved CAR T-cell product, which is ide-cel, and another one, which is cilta-cel, is that this bb21217, although it targets BCMA, what we do with this drug product is expose it to bb007. This is a PI3 kinase inhibitor. And the idea of exposing it to this PI3 kinase inhibitor in vitro is to try and enrich for a memory like T-cell phenotype. And by doing so, the idea here is to improve upon the duration of responses that we're seeing with drug products such as ide-cel. So that was the goal of this study, and that was the rationale for doing this trial. What we have here is updated results on 72 patients. If you look at the overall response rate of this patient population, it was close to 70%, so pretty similar to that seen with ide-cel. Complete response rates were seen in about 39% of patients. But once you get to the recommended phase 2 of 450 million cells, the median duration of response has not yet been reached. We see that patients have a duration of response overall in patients who have an enriched memory like T-cell phenotype as defined by increase in CD27. It's about a little over 27 months, so certainly better than what was seen with ide-cel. And obviously, we need longer follow-up on demonstrating this. What we also will be presenting at this year's ASH is the number of CAR T-cells which are detected at year 1 and year 2, and that is in fact a predictor of duration of response, at least with these drug products. And what we're seeing is even at 2 years, you can see circulating CAR T-cell products, so suggesting that T-cells are persisting for a little bit longer. So the hope is that with this exposure to bb007, with enrichment of this memory T-cell-like phenotype, we are seeing persistence of these T-cells last longer, and this will then translate into an improvement in duration of response as well. As far as toxicity was concerned, there's no real difference between what we're already familiar with. So CRS was noted, very little neurotoxicity was noted, but most of the toxicities with bb21217 were also very manageable, as we become used to seeing with these CAR T-cell products. You know, I do think we want to see the data cut off at a longer time point. For the early data, which we will be presenting, the fact that we've gone out to a duration of response of close to 27 months, we've certainly done better than the 20 months that we had earlier with bb21217. So that looks good. And the fact that we are, with this intervention of the PI3 kinase inhibitor, seeing an enrichment of memory like T-cells, and the fact that they correlate with response is all actually favorable. What we will need to see and do is how can we try and make sure that the majority of patients have this benefit of the enrichment of the memory like T-cells. And for that, I think there's ongoing work looking at the initial T-cell fitness of a patient and who are the ones who benefit from this kind of an approach to start off with.